When Ron Xiaowei and I were thinking about how to sort of organize the session, we identified sort of three major domains of technology that are sort of driving measurements of biomolecules in tissues. And the first is using antibody-based strategies to bind to mostly obviously to proteins. And the second is to use in situ based hybridization approaches to probe mostly gene expression or features of DNA. And then the last is sequencing based approaches where material is captured from tissue in some sort of spatially resolved manner such that you can identify those biomolecules in space using sequencing. So we thought that because each one of those different modalities of measurement have unique challenges and opportunities and applications, that it would be best to split them up into those three different areas. And then in each individual area, kind of identify the respective opportunities in those different areas and specific challenges that those particular modalities of measurement really have.
Good. Thanks.
Sure.
Since we only have, say, about a dozen people, one way to encourage others to speak up is probably let others to introduce themselves. Even
I've been trying to, but I think the thing is that the individuals who are muted and have their video off are participating as passive participants. So I would encourage any passive participants who feel like they can take the plunge and talk with us directly about these challenges to unmute your microphones and turn on your video.
Hi, this is Chengyan Lo from UCLA, Department of Human Genetics.
Hi, Chengyan.
Hi.
How are you?
Good. Yeah, we have been primarily working with single-cell DNA methylation profilings. And generally, I'm sure a lot of groups are heading towards that direction. At present, there hasn't been a lot of epigenomic profilings in the spatial techniques, like similar to the ones you're working on. So if anyone in this group are developing that kind of assays, I certainly will be interested to hear about it.
Yeah, I think that would be a really, really exciting, I think there's a lot of exciting opportunities there to sort of extend to different modalities of measurement. And I think certainly DNA methylation would be a really, really important one.
I have a question.
Anybody else want to? Go ahead.
Oh, sorry. Well, let's other people.
Well, I'm just trying to corral maybe one or two more people here.
Okay, yeah.
Is anybody else joining us?
Christina, I see you.
Hey there. Yeah, sorry, Christina Curtis. I'm at Stanford. We're broadly interested in spatial technologies and their integration. I would say on the sequencing front, have been doing some work with spatial transcriptomics and quite a bit more on the spatial proteomics front. But we're very interested as these next generation of tools come into being and really advance to be able to leverage them. So happy to
Awesome.
do that.
One last call for other active participants in our discussion. Anybody else who feels like they would be able to participate? All right, so if you have a look at the discussion prompts that we kind of came up together, the moderators came up together, and I'm happy to start in any of these particular points. We have to have a scribe. Somebody's chatting here, but is anybody willing or interested in scribing for us, just writing down some of the notes, the points that we capture? I can also try to do it while we discuss, since we have a small group. All right, I will try to serve both roles the best of my ability. So, you know, sort of the first thing we had sort of wanted to kind of talk through together as a group is, you know, what are the methods that are available right now for spatial measurements in, that use sequencing as a readout. And, you know, maybe we can start with, for those who are here, you know, what techniques are you using, or what techniques are you most excited about using?
So I'll start. I'm aware of course, the published work for merfish and thickfish. Also aware of commercial offerings, Visium as an add-on in the Chromium system, but also this year we heard more from, blocking on the name, but at the recent LGBT meeting, there were announcements of 2 or 3 platforms. NanoStream would be one.
Mm-hmm.
That was a conversion of the physics from the George Church's group, but I forgot the brand name. These are sequencing based, so I'll start from there.
Yeah. Oh, somebody's scribing. There you go. Thank you, mysterious person. That's right. Yeah. So somebody was just pointing out that the method you were describing, the in situ based approach that
Right.
originated from physics is by a company called ReadCore.
Yeah. Andrew is scribing, and he.
Yes. Thank you, Andrew.
ReadCore. That's the name.
Yeah, and please don't be shy. You know, we're a small group, so if anybody else would like to participate, please do. Christina, do you have any other thoughts on particular approaches that you're especially interested in or using? You said you mentioned Visium, I assume.
Yes, we've done just a little bit with that. We've worked with NanoString on the digital spatial profiler, which has the dual protein and RNA readout, not on the DNA front yet. And then we're also quite interested in these other approaches, merfish and seekfish, both from the transcriptomic, but also the DNA-based readouts as well. And so I think I've had to play with a lot of them, but fairly early days in terms of the data being generated and really thinking about methods. And then separately, we also work with folks who are developing MIBI and some of those methods.
Right. Okay. Yeah, we don't, I mean, I think MIBI is probably, well, maybe antibody based. So as opposed to sequencing based, but
right.
Yeah.
Separate chat room somewhere.
Yeah. So I think just the other method, just to make sure that we include, are obviously Visium here on the top under ReadCore, which is /spatial transcriptomics. I can type it in. And then our method, which we developed last year, called SlideSeq, which is in many ways similar to Visium. It captures now at this point gene expression data, but it does so using barcoded beads that are much smaller than the spots of Visium, which are 55 to 100 microns, depending on how you measure, because there's 100 micron center to center distances, but 55 micron diameter spots. So the resolution is somewhere in that range. And then NanoString's resolution is, does anybody use NanoString? I haven't actually used it, but I've read a little bit about it.
Haven't.
Yeah.
How does this work? Can anyone hear me?
Yeah.
Oh.
Hi.
I think it goes down to 1 micron, doesn't it?
Yeah.
It has it uses these mirrors like you have on a laser projector that are 1 micron squares.
It's very clever, yeah. So you can capture individual 1 micron squares. There have been some, as far as I understand it, there are some challenges associated with actually recovering material from such a small piece of tissue, right? That you have some kind of dead volume in the course of the machine, such that even though theoretically it's 1 micron, you wouldn't get very much material from that 1 micron.
Yeah,
but it's,
I think Evan, I think you're right. Sort of for the small regions, maybe it's not so good. I think what appealed to me about it is it's completely flexible in the region you want. I think at a clinical level, it's actually pretty neat. You can just define anything at any scale at fine resolution is unique.
Yeah, and I think another really nice thing about it is it has a kind of the workflow and the thought process is kind of similar to an older, sort of laser capture microscopy modality where you're sort of physically looking at the tissue as one would as a pathologist, for example, and sort of taking regions of interest, and one can easily kind of combine this with histology data, for example, to find regions of interest that a sort of traditional pathologist might identify. So there's a really nice kind of synergy and sort of extension of existing workflows for laser capture microdissection. I think another thing that is interesting about it with respect to the other methods is that it appears to be quite compatible with paraffin-embedded formalin-fixed tissue. So that's something else we can discuss too. But I think a major challenge of these different methods, and this is a challenge that really extends across all three different subgroups, is what kinds of tissue can we really be doing these assays on? And right now, one really major advantage, I think, of the NanoString platform is the ability to generate decent data from paraffin-fixed, formalin-fixed paraffin-embedded tissue.
I was interested in Christina's comment about DNA. I think somebody else also mentioned that. I think that seems like a new area of interest. So all I've heard of before is like transcriptomics and maybe some protein, but interested that people are starting to think about DNA.
Yeah, I agree.
Yeah.
Sorry, I'm trying to get my phone to behave, but it puts me in some other chat room. So I had better connectivity. I don't know where I am there. Anyways, I think people have been trying to sort of scale the ability to look using fish type probes at copy number. I mean, there's also been other ways to probe single mutations, but the challenge is around the scalability of these methods. And so we're quite excited about where that will lead in terms of having spatial resolution on those data types, but I think it's early days. It's also not my particular area of expertise. I don't know, June, if you've seen some of that coming along as well.
So we've heard a few presentations about ReadCore. I think it is public knowledge that they are it's a single molecule in situ sequencing. But it's not whole transcriptome, it's panel, like 200 gene or so. Then you do need to have a lot of upfront informatics work to have one panel, say, for the nervous system and other for immune system. There is a lot of potential to use the same barcoding system to do multi-omics. So allocate that 200, some for transcript, some for proteins. The demonstration data, we currently don't have access. We see proof of principle data. It can do X, Y, Z, 3 axes, although the resolution are not the same in all 3. I think the field of view is good. I don't know how to quantify that, maybe hundreds of cells. The software, as you know, in any of these companies, is a fast-moving kind of a process. I think the users are most hungry for some automated software to segment the images to identify cells, and then to call the cells by referring to reference transcriptome. So those are currently not quite turnkey. That's my impression. It could be wrong.
Yeah, I think you identified one really important challenge of all of these methods, which is I'm just was sort of looking, maybe Andrew, you can find the appropriate subtopic point where this could be described, but I think the question of cell segmentation is one that's really key, and it's something that's relevant, but the challenges are specific to all and unique to all of these different modalities of measurement. In the case of an optics-based approach like an in situ sequencing or FISSEQ, the challenges are more about segmenting individual cells from images. In the case of these transcriptomic-based strategies like SlideSeq or Visium, it's about trying to sort of deconvolve complex mixtures of cells that are contributing to the RNA or the DNA on a particular spot, right? So they're both computational challenges, but they're kind of challenges that are quite different.
Right. So I do have a question both for Evan and for others knowledgeable in the room. That is about benchmarking across different technologies. Everyone might say it worked in my hand, but these are data with different design. Let's say if you have a 200 gene panel, you design your probes differently than somebody else's 200 gene panel. It would be nice to plan a kind of a multi-platform. People just do this comparative study, say go to a system where it's well known, whether it's hippocampus or the retina, to show you can identify the same 2, 3, or 20 cell types everybody else has published on, and know the biases of your system to know which cell types are particularly elusive in your hand or which are really good in your hand. That requires a lot of coordination, I would say, to do well.
Yeah, I like your idea of the retina too, because so many years of study have given us a really nice sort of spatial layout of cell types, right? So we have an expectation not just from a lot of the single cell data that's been generated in the tissue, but also about their spatial location. So both of those things I think could be great ways of kind of validating. So I mean, I think that would be a great thing to sort of dig a little bit more deeply into. You know, when we say validating or comparing, it's good to be specific, right? So like one thing I think you mentioned, which is like, you know, how well do you discover cell types, right? Is a really key question, right? How well can you sort of identify specific cell types that maybe are identified as in the reference from a single cell data set? Are there other things that people can think about that would be sort of valuable benchmarks or evaluations for data? Because I think it's not just that, you know.
Yeah, I mean, I think dealing with a different, if we're talking cross-platform, which is a little bit of a different issue, is, you know, dealing with differences in resolution and sampling, and how do we embed that sort of fine-grained mapping when maybe you're, you know, I mean, you could envision in an ideal world, maybe you're running a lot of these technologies on just adjacent tissue slices, right, that are off. Still, we would need to register those, and I think that's doable. Most of these are pretty thin slices. But I guess something that certainly we're hoping to learn from these and other methods as well is to put that in the context of the tissue architecture itself, and maybe having some fiducials can sort of help with those, you know, how much you want to sample. I think that was the point that was brought up earlier, especially with respect to clinical samples that can be incredibly heterogeneous. So different challenge, but maybe fits in there somewhere.
So like a registration to a common coordinate framework, you're saying?
That's right.
Yeah, I agree with that. And each one of these strategies definitely has these different measurements has different kind of challenges associated with you know, registering to the CCF. Mm-hmm. Yeah, I mean, I guess the other thing I was thinking about in terms of comparison is. Well, I mean, I guess this is sort of its own domain, but it's the question of resolution. You know, for what kinds of problems, you know, obviously resolution can span in these measurements. It can span, you know, from tens of nanometers or hundreds of nanometers all the way up to hundreds of microns. So it's useful to think about where and what magnitude do these different spatial resolutions matter? Anyone have any thoughts about that?
I think at the clinical level, it doesn't matter to be that high resolution. At least that's what I hear from people, right? From the pathologists, they care less. Maybe for for real understanding of cell types, you've got to get down to the lower resolutions. But if you're just trying to say, use it as a method to separate out tumor from normal, you don't really need to go that lower resolution. Is that what you're getting at?
Yeah, I'm just sort of thinking like, you know, when, in what, you know, for those who are using these methods, you know, how do they see, you know, spatial resolution mattering, and sort of where are there particular inflection points for certain kinds of questions? Like, oh, you know, in this case, you know, 10 microns versus 100 makes a huge difference, or, you know, 100 nanometers versus 1 micron makes a huge difference.
I mean, I would agree with what was just said, that I think in a lot of these clinical tumor applications, we have such a limited understanding that I think even being able to map at a pretty granular way, what is the tumor immune contexture, that gets you pretty far in terms of being able to appreciate that these diverse cell types are present. And, you know, there's other questions where you would maybe want to even have a single proper, you know, greater resolution on the question, but I think it does depend on how deep you're trying to dive into the biology. And if it's sort of for biomarker discovery, not so sure that that's absolutely required. And when you think about the path to clinical implementation as well, that's presents some other challenges in terms of what would be required there.
Right.
I always view the ultimate goal of spatial analysis is to replace, not just being complementary, but really replace dissociation-based single cell analysis. And I really think this single cell resolution is going to be critical. But a challenge there is a lot of time for mouse tissues or smaller tissues, we can more easily perform these unbiased sampling across the entire organ, for example. But when we really tackle human tissues, it becomes a challenge. So it's very unlikely you can actually sample the entire human organ of any sort. So there you're going to have to deal with these two levels of heterogeneity, both at the tissue level, and then we'll want to dive down into a single cell resolution.
Yeah, spatial coverage, I think, is another really important domain comparison, right? So for example, I mean, I think that's something we were talking about, the nanostring platform. That's an example of where it's very challenging to use the nanostring platform if you really need that kind of large scale three-dimensional reconstruction, because you have to go in and with your mirrors and identify every little spot. So unless your spots are really big and very close together, it's very hard to assemble, you know, an entire sort of surface area of tissue, whereas some of the other strategies are more scalable to larger numbers of sections. And actually, really, I think for the sequencing-based, the sort of Illumina sequencing-based strategies, it's really about cost, that, you know, the cost of the sequencing that ends up being the prohibitive thing, not other aspects of the pipeline.
So Evan, I wonder if you can say a few words about de novo sequencing, such as SlideSeq versus the current state of ReadCore, which is really a barcode sequencer. What do you see the immediate kind of future? Are you saying these two kind of platforms will go in parallel for a period of time, or you see the limiting factor for de novo approach is really the cost will eventually get too high, or you think that will be okay?
Yeah, I mean, I think, so I think you're drawing a distinction, just to be clear, you're drawing a distinction between direct sequencing of the actual material, the RNA or the DNA that comes out of the tissue, which is how Visium or SlideSeq works, versus sequencing some sort of proxy for the hybridization event, which is kind of how the nanostring platform works, where they hybridize these probes, and there are many, many probes on a particular transcript. So then those probes all bind to the transcript, and there's a photocleavable barcode that then gets released and then is sequenced. I thought ReadCore, I mean, the whole point of FISSEQ is to directly sequence the material. I didn't realize that ReadCore was moving to a hybridization-based strategy. Is that right?
from the AGBT presentation, the current read core seems to be a barcode sequencer, although they could certainly extend to, well, it's a padlock probe, so I guess currently they're sequencing the barcode, but they could totally also just sequence the part that replicated from the RNA, I suppose.
Yeah, I think that's correct. They're currently sequencing just barcodes, but they claim that they have the ability, you know, as was from the original paper, I think, to do actual sequencing of all transcripts. But right now it's just it's like, I think it's similar to the STAR-seq, the
star map. Yeah.
Yeah, it seems to be similar to that, the approach. It's like a MIP-based.
Interesting.
I think, yeah.
I wasn't aware of that. Yeah, so just to answer your question, I think, you know, one of the, and this is a strength of all hybridization-based strategies, which is that I think one of the major strengths is the tissue types that they would be most easily deployed on, because hybridization approaches tend to be more resistant to, you know, tissue damage fixation problems, whereas sequencing-based strategies, you have to do enzymatics on the material that you take out. So if it's been, if their adducts have formed using, you know, from formaldehyde fixation, or the RNAs have broken up from being dried out for long periods of time, it can be challenging to deploy those on such sort of beat up tissue or preserved tissue. Obviously, the advantage, I think, is in the detection sensitivity. You know, the alignment to the genome is obviously like or transcriptome is obviously a very, very, very high quality confidence that you actually have what you think you have there. Whereas there are challenges in some of these hybridization-based strategies with false positivity. And some of this has been mitigated. Like NanoString's approach is nice because you have these 10 different probes on an individual transcript. So you can set thresholds for confidence, right? If you only see one or two of them, then probably that transcript isn't really there, or, you know, it's binding to something else. But nonetheless, it can still, it still requires some kind of, noise model to deal with some of these issues. So I think those are the sort of pluses and minuses. And I think if your tissue is good and it's frozen, there's really no replacement to being able to get the actual transcriptional, transcriptome data. Obviously there's also the resolution question with some of these strategies as well. But yeah, I think that's sort of the most important sort of way of framing it. And I would say, that, that although we are increasingly being able to multiplex more and more these hybridization-based approaches, I think the thing that we see as so valuable with the sequencing approach is the discovery. We talked about, biomarkers for clinical samples, for example. I mean, I think we've perpetually been surprised by the kinds of genes or programs that are, expressed in certain tissue types. And so there are many things that you wouldn't necessarily be looking for. And it's always interesting and potentially very powerful to be able to find those things in the actual tissue samples. Does anyone have any other thoughts about that?
About resolution, I'd like to follow up on the discussion. I feel it is very application dependent. If you just speak from first principles, there are those who care about subcellular structure and function, say RNA organelle or condensate in basically the function of different organelles. They couldn't care less about a larger field of view. But then many of us, especially for clinical samples, we do want to get a single cell or even one level above. It's like a regional heterogeneity in a tissue slice. So to define this in terms of application domains, let that dictate the scale. I find it useful. I see in the comment or the question by Simon about whether there is a system that offers flexibility, so you can do both global view discovery, but somewhat sparse, but then zoom in to higher resolution, but targeted. So trade long and short to a short and long. I think that's indeed the dream. I've heard a talk about microscopy. I'm just paraphrasing by secondhand knowledge. They refer to the dream setup is what's called a Swiss Army knife of microscopy. So there are modules you can bring in, one for the broadest view, but not very high resolution. Then you swing in another that can be super high resolution onto subcellular, or you can do another one with low illumination, so you can image for hours or days, even on live samples. That type of flexibility, of course, is the aspirational goal. Most of us would be happy to get a toehold on even one of those.
Yeah, I really like that question. It's sort of when we have this question, what are the paths towards measuring multiple modalities? In some ways, that is multiple modalities, even if you might be measuring the same biomolecules, but it's a way of it's sort of a hybrid discovery and targeted based approach. I think that's a really interesting idea. Let's see, what other...
I have one final question. What do folks think about as the single cell data that we get from the traditional methods increases, doing deconvolution on broader areas of spatial data, and whether that's going to solve the problem? Or...
Yeah, I'm quite confident that in the afternoon session on spatial profiling tools or computational tools, that's going to be either the number one or number two question. People have different ways of describing it, mapping or projection or deconvolution, but definitely I think that is an absolutely key area. And probably the strategy is going to be different, obviously depending on the resolution, right? Because the computational problem of deconvolving a 100 micron spot versus a 10 micron or single molecule data is very, very different. Here's a question that I thought we haven't really touched on yet, but what improvements are needed for optimal deployment? Like, what do you guys see as the sort of biggest challenges to getting the technologies that you care most about sort of used in your labs or in labs that you work with?
So in our medical school, I think there are different types. There are medical science researchers who say, oh, you have a vision. I have sequencing dollars to burn, but I don't have anyone to analyze the data. But let me get the preliminary data in 3 months. Let's see what is the company offered first look at the data. Others are more kind of deliberate, wanting to get an informed comparison of the 3 or 4 platforms before deciding. But then it's right out of the gate, the choice between commercial offerings, either locally or outsourced, to the homegrown ones. Then you need a super postdoc to really do it. So these are the choices that people are grappling with.
Yeah.
Yeah, I mean, I would just echo that. I think as someone who's interested in also developing methods to interpret these data, a lot of it comes down to, can we get our hands on some of it so we can start thinking about the analytic approaches that might accompany these tools. And, in some cases you hear about these things coming and it takes a while unless you really have drummed up that collaboration early on or are waiting for it to come down the pipeline. And I will say, that's influenced some of our choices in just trying to get a handle on different flavors of data and what's accessible so that we can start thinking about that type of integration. And yet each technology has its own nuances. So it's not that they're all necessarily generalizable. I mean, of course, there's some principles, especially as you get further downstream, but really for the raw data, it can be quite different. So
to echo what Christina said, for instance, if we have say vision data in hand on a well-known tissue, let's say retina, Evan, you mentioned, or to hippocampus, many of us have published on, we will be able to know how easy it is to deconvolve the data for that neighborhood into known cell types in the Atlas catalogs, but knowing that the probe behavior will be very different from the Atlas platform to this imaging platform, there might be just real hurdles that preventing you from successfully deconvolving into known cell types.
And I think I guess I would just add, speaking from the sort of NCI-HCA efforts, there's of course a lot of interest in trying to have not necessarily all of these tools that we've talked about here, but to generate some reference sort of data sets so that people can begin thinking about deploying the computational tools. And I guess I would think that this could be a broader goal of looking across atlases. You know, it does seem like certain atlas efforts, because of the know-how of the investigators that are involved, are focused on different technologies. And, you know, in fact, for this cancer atlas, right, we're doing a lot of work with thymus and other tissues just to have really a reference that will be valuable across comparisons. And I would love to see that extended, because I think there's groupthink that could be applied, and just having access to the data. So I don't know, that might be some action item that comes out of this, to learn what other atlases are specifically doing with regards to these technologies. I would love to sort of see a compendium, and that's a moving target, but certainly would be interesting to know about.
Yeah. I mean, one major advantage, I think, of these kinds of data is the fact that they are sort of most similar to single cell data sets. And so presumably many of the tools that have been built in that domain may be obviously not immediately relevant, but potentially relevant computation.
So Evan, looking at your document, are there other questions you think we should turn to?
Yeah, I'm just flipping through right now. I mean, we had a little discussion about capture efficiency. It's a little bit of a technical question. I don't know if folks here want to discuss it, but do you guys have thoughts about, you know, how important it is, capture efficiency or detection efficiency?
We are already in the company show and tell. We sometimes ask whether the most abundant molecules you already suffer from optical crowding, but for the low ones, you don't just see them enough. How to balance this dynamic range is a question that's very different for different platforms.
Right, for sure. And then I guess the other thing is, you know, sort of the final question there, thinking about multiple modalities, especially since Chungwan brought it up at the very beginning. You know, what are some particular modalities of data that people think are most important to add to, you know, let's say to transcriptional data, since that is the sort of dominant measurement that we are currently doing with these sequencing-based strategies?
I guess typically a taxi would be more easily achieved, right? Just like in the case of a joint profiling of RNA, a taxi was relatively straightforward in the dissociation single cell. I would kind of imagine that might happen first in the spatial space too.
Yeah. I mean, another thing, the other thing is obviously protein. I mean, I think one thing that's often not quite appreciated, but now with some of these techniques like site-seq approaches that allow you to combine single cell RNA with measurements of specific, you know, antibody-based measurements of specific proteins, is that the gain that you get with repeated translation of the same transcript is quite remarkable, and in the sense that, like, your detection sensitivity for many molecules is way higher in the protein space, just because there are many more copies of the protein than there are of the transcript. So that can be very useful for, it's useful in single cell data. It can be very useful in spatial data, which may be sparser. Anything else anyone wants to add in the last couple of minutes?
Question, where am I supposed to stick the terrible notes that I made?
Oh, I was trying to jot down some notes because I wasn't sure. It's in that green box, if you recall the slide that tried to hit us over the head with it.
Green box, green box, green box. Is that that's in the web thing, right? Oh. Where on the Google Doc where it's the spatial breakout discussion prompts?
Yeah, I'm gonna just put this in the chat box just in case you don't have it. That's the link. And then
Okay.
there's this green box, and I've just been kind of taking some notes. So if you could put your notes in there, that would be quite a relief. So I don't know what the acronym is for being harangued or whatever.
Everything I type, spelling errors, a lot.
That's fine. That's totally fine. The second part of this this discussion will be really very focused. And they've allotted a very large amount of time, 75 minutes, but I certainly don't think we would need nearly that much time, where we will just be trying to identify some very specific areas where collaboration could be useful in addressing some of the problems that or challenges that we brought up in the first part. So I think some of the things that we brought up in the last session were very clearly geared towards collaboration, where somebody, for example, mentioned the challenge of data generators finding analytical tools or analysts trying to get their hands on specific kinds of data. But maybe we can formulate, something like the goal is 5 of these individual sort of collaborative efforts that can address some of these challenges. Is there anybody else joining us who didn't join in the first part?
Yeah, this is Ajay Pillai. I'm from NIH-HCA. I'm part of the HubMap program.
Hi.
I was in the antibodies discussion for the first session.
Okay, great. Yeah, and when Ron and Xiaowei and I were coming up with the discussion topics, many of the topics are very similar. It's just particular nuances and challenges for specific data types. So great, nice to have you with us.
This is Rahul teacher from the New York Genome Center. I was in the MERFISH and imaging-based panel earlier, joining here now.
Okay, great. All right, great. So let's see if we can tackle this last part. So if you look at the Google Doc, you'll see that we've come up with kind of my and Andrew's joint scribing has kind of come up with a set of notes from our discussion in the previous round. So it's a little bit disorganized at this point, but hopefully we kind of have an overall sense of some of the things that we talked about before. Does anybody have something that they'd like to nominate as one of the sort of 5? So if you look in the breakout session 2, you can see it says, capture a minimum of 5 recommendations for collaborations that might address the problem or questions that we talked about before. Does anybody have something they'd like to nominate as a first thing to discuss? Does this make sense to people? Does anybody have any questions about it? All right, so I think one thing that came up as a major problem, and it obviously sort of delves into questions about computation, which might be addressed more later in the session. But one thing that I think came up as sort of a challenge for all of these strategies is sort of data integration, right? Integrating data from multiple modalities, but also sort of evaluating how, methods for how to integrate data together. So do we think that's a reasonable way to, some approach for sort of encouraging collaboration?
Evan, you're talking about different modality like RNA to ATAC-seq.
Sorry, no, I meant actually, I should have been clearer. I meant, different measurement strategies.
Like a comparison between platforms?
Yeah, exactly.
Yeah. I think that's quite important. I guess a lot of people who come to this room have the interest to learn the latest about which are more ready than others and their strengths and weaknesses. I didn't attend some of the more recent meetings and feel wanting to hear the latest knowledge percolating to our way.
Okay. What are some sort of things that we think would be particularly valuable in a collaboration where we're trying to compare across platforms? I mean, certainly we want to have very clear standards about, so one thing you mentioned before, June, was a sort of a canonical tissue, right?
Right. So even the basics, if we put 4 or 5 up there on the dashboard, we don't quite know. Well, we need to agree on which 3 or 4 or 5 yardsticks we measure them with. We talked about resolution. We talked about number of features. We talk about flexibility. There might be cost. There might be whether they have a Z dimension or not. Whether it's easy to get it to work after a few tries.
Yeah.
These are kind of the immediate yardsticks I can come up with.
Compatibility across tissue types, right?
Right. And then what you were alluding to is this difficult task of comparing across platforms, because sometimes when we ask, hey, how do you, of course, if you say this is important, we know that's true, nobody will disagree. But what is your advantage to your closest competitor? Sometimes the answer is, oh, we do this, nobody else is doing this, so I have no way to benchmark, we haven't tried. I find that answer somewhat unsatisfying, because for the basics, let's say, go to Retina, can you see the 5 major cell types? Show the data to show you can do that, not setting the bar too high for, say, 100 cell types, just show you, go to hippocampus, show you can distinguish excitatory and inhibitory neurons. So that'll be some kind of a benchmarking effort that requires us to share data soon, I guess.
Yeah.
I like the idea of having benchmarks that are biological in nature in addition to technical in nature. People get so caught up on technical metrics and resolution and genes per spot, and all those things are important, but they can also be misleading, because you can measure lots of genes that might actually represent background as opposed to real signal, and your resolution can have bleed through issues, and so what June said about sort of having sort of yardsticks that you have to reach biologically. Well, can you discriminate X versus Y, and then can you discriminate subtypes of X, and can you get farther? I mean, this is how we often evaluate data sets that we see, right? And I feel like that's particularly important in spatial, where we do have some good senses of the types of things we should be hoping to see, at least at a cell type level.
Yeah, I completely agree with that. And I think what's really nice about these biological, we can even be very specific if we want about the kinds of biological benchmarks we care most about, but we can even extend it and say, for each one of these benchmarks, there's a technology that kind of detects it best, right? Or does the best job. So for example, maybe we define, 5 or 10 markers that we have measured by a very, sensitive single molecule fish strategy, in series. We have certain very clear, we have histological stains that give us very clear understanding of, overall macro architecture of tissue. How well do those particular features get identified in these other modalities? That seems like, it seems like we could maybe have these kind of like reference data sets with sort of like the best in class tools to measure those features.
Yeah. I'm also trying to think more concretely. Imagine somebody give you a data set saying, here are 1 million molecules measured in XYZ coordinate. And then you know those 1 million came from 200 different genes that's in your panel. But one of the first things you want to do is to segment them into cells, but then get the cells vector, then see how they can be assigned to known cell types. I've yet to see a good example or a quick way to turn that data and map to the catalog to identify even at the base of the tree 4 or 5 cell types to show their work. So maybe you guys.
Are you saying that you're saying that you haven't seen examples of people mapping single cell data into spatial data? Or I'm not sure I quite understand.
Right. So at mid distance, I don't have the proximity to the latest data. So I'd love to hear you guys who have the newer data to refer to some examples where your spatially located cell, you got that cell's gene expression profile. Knowing the quantitative measurement is different from the drop-seq or those in the catalog, in the atlases. What are some successful examples where your cells in situ can be reliably mapped to known cell types? There must be good examples. I'd just love to learn more. Like Rahul worked on lung map. I guess there must be examples.
So I think, both Evan's lab and my lab last year, and since then, a number of other labs have shown that it's possible to take in situ measurements from technologies like StarMap or spatial transcriptomics or MERFISH and map them onto single cell RNA-seq catalogs. I actually think that is, to me, one of the most important benchmarks is how well can the data from a spatial assay be mapped onto a ground truth list of cell types, in part because that's one of the goals that we want to achieve anyway, is to always think about a tissue in the context not of what are its broader spatial patterns generally, but really what are the pieces of the puzzle that make up the tissue, the cells themselves. So I think there's been tremendous progress in that area, and I think that's a very valuable benchmark that people can apply. I wanted to ask, I wasn't here for the first part of the session, but I know that the Allen was doing really kind of exactly this. They had this, what was it called, Space TX, I think, where they were collecting lots of different technologies on the same sort of canonical tissue, even with the slices being prepared in an identical way. I don't know whatever actually, I haven't heard much about what happened with that effort or how that can inform some of the path forward.
Did you want me to say a couple words about that?
Yeah, I was just going to say, I saw Amy here.
Yeah, hi. So actually, the best person to talk about this would be Ed Lean, who's currently in another session that is also talking about this. I've actually been skipping around some of the different sessions and there's a lot of overlap. So regarding the spatial transcriptomics, we're still working on it. We have made, I guess, a lot of progress, but we still have technical challenges, particularly when we're looking at the human tissue. So mouse, it's been a lot easier for us. But we are, our main focus actually has been on scalability. So I would say on a sort of bespoke small scale level, we're doing all right, but scalability is still a challenge for us in terms of robustness of pipeline development. So yeah, that's kind of the status. And there's a bunch of people that have been working on that associated with the BICCN project, Brain Initiative Project, is kind of where that's been fitting under. If people have very specific questions, I can answer some of those, but I don't know exactly, other than yes, we're working on it. And it's in terms of our benchmark standards, it's exactly as Rahul said, it's basically mapping back to our, kind of keystone set of known cells that have been profiled transcriptomically.
I also like, though, do you know whether Space TX has been thinking about other specific kind of biological metrics beyond just the single cell inventories? Because I think that's a really key point. It's something that we really care a lot about in our tech dev work is, what histological features can we sort of use as benchmarks? Are there specific, gene expression patterns that can be kind of used as ground truth? So I think, I'm just wondering, do you know sort of if there's like kind of a list of things and there's like kind of like a performance tab for each one, or is it just the single cell?
No, there's a paper that we're like literally days away from getting into bioRxiv right now, where we're essentially using spatial transcriptomics alongside with electrophysiological recording and morphological reconstruction. So that's what we've been doing, at least looking at cortical cells. That's kind of building on a lot of the data that we already have in our single cell data set that we've been literally building for over 5 years now.
Yeah.
And so one of the benchmarks specifically for that is to go through methodologically, tag individual cells using a patch-seq technique. So basically doing the electrophysiology, like Tolia's lab has been doing this for brain. Electrophysiology, morphology, nuclear transcriptomic profiling, and then also correspondingly mapping that back onto a spatial transcriptomic sort of reference point. So this is where we've had success doing this in a sort of, again, kind of bespoke way, but for the spatial transcriptomics for scale, it's not, we're not there yet.
Mm-hmm. Mm-hmm. Okay. Yeah, I mean, in some ways, SpaceTX is like a comparison across not just the sequencing based, but it's obviously a comparison across all three sort of domains that we are discussing in these individual subgroups. But I think it's nonetheless still valuable to mention it because that information is not percolated into the community, and there may be ways in which we need to extend or refine the results of that initial survey beyond. So I think it's important for us to kind of mention this comparison as being really important.
So I had actually a question related to that, because certainly for brain, we have some pretty obvious ways in which we've been taking to approach looking at electrophysiology. So looking at properties that we can ascribe to very, functionally direct or obvious attributes of cell function. And I think one of the challenges that I'm sort of curious about some of the other consortia associated with HCA and HubMap is how accessible are some of those? So one of the previous sessions I was just in that actually closed down because there were insufficient people in it had to do with looking at other attributes of non-transcriptomic based cell identity sort of cross correlation. So how well are other modalities, whether you're looking at cell surface proteins or if you're looking at, I don't know, like some other functional output or behavior of cells. In brain, we're looking at electrophysiology and morphology for those. You could look at neuropeptides, you could look at any number of other things, but how are those quantifiable other linking points that would complement looking at spatial transcriptomics being parsed through the other organ systems?
Right. So you're asking, are there other examples in other tissues where, I mean, of course there are, but yeah, what kinds of
the standardization there?
Yeah, the standardization,
the integration, and then the metadata that makes that scalable.
Mm-hmm.
I almost have a question, both a comment and question to Amy, that for the brain, I guess, other than the ones you mentioned, electrophysiology, morphology, and single nucleotide RNA-seq, you must have also been looking at localization in the laminal and aerial columnar sense, but also the long-range projection, the connectome type of work, right? Do you also score brain region, almost like the Enigma consortium, the cortex architecture? And then there are those who turn from morphology to force, like the cell is a stretched lung that must be experiencing stretching force. Are those modalities you are doing with Brain Atlas?
The short answer is as many as we can. So obviously with human, we're unable to get full long-range connectivity when we're working with human excised tissue, that's surgically excised tissue. We do pin essentially local fiducial reference points. So we pin certainly the nuclear localization with respect to, and then the immediate morphological reconstruction that's local to that within a cortical column. And that's data that we capture, we actually put online as well as metadata. For mouse, we have been doing that. We've been doing a larger cell reconstruction in terms of projectional mapping in a much, again, lower throughput way, where we're able to do that a little bit through looking at transgenic lines and then linking up again transgenically derived cell types that can, that we essentially pin to their family class or family cluster using other metadata as well. But that's a much more painstaking way. So we actually have a number of papers published now that are trying to kind of aggregate together the critical sets of metadata that give you confidence of that cell type identity, looking at those sort of key factors or attributes that co-cluster along with the multimodal elements that are not just relying on a readout of gene expression for, you know, the RNA mapping alone. But that's again where it comes back into this question of validation. What are the best validation points when you move back into spatial transcriptomics that are sufficient and appropriate for a given cell type? And, you know, if you're just working with, you know, some UMAP cluster analyses that may not, we've demonstrated and others have as well, that that's not going to necessarily give you the most functionally salient cluster definition. But this is very iterative, and for us it's slow. For other people I imagine it is as well, and it's very much of a, you know, test and iterate. And so that's where I'm kind of curious about where the other fields as well are finding those kind of key functional points that are going to reinforce what are those appropriate either marker groups or subsets to be looking at when you're doing spatial transcriptomics.
Yeah. So maybe something else, which again, it sort of transcends just this group, but maybe something else that would sort of be a key point of collaboration is kind of, you know, these HCA meetings have a lot of like breakout groups that are very tissue specific for various questions that are sort of the main topic of a meeting. But maybe one focus really could be on, for each tissue, sort of establishing some of these like core principles that would be useful to sort of use as benchmarks, right? Like the way that, you know, we were describing that, you know, we know in the brain that, you know, you can't have, you know, excitatory neurons in layer 1 and all these other kinds of like basic principles that, you know, that's just a very basic one, but obviously these principles that kind of define how you know if something's working in the brain. It may be very useful for technologists to have that information. I would certainly love to have that information in other tissues as well.
That's a great idea. That would be really valuable.
I think another example for something that could be valuable for technologists is those types of principles, but especially where they're very quantitative and relate to the possibility of detecting bleeds through or limits of resolution. So I remember seeing you talk about SlideSeq, Evan, that there were a couple of examples where you knew, for example, that there was a structural feature that was of a particular width. And then you could go back and look in your data and see, you know, how far did that structure extend? And you could use that to benchmark whether or not you had any bleed through from one bead to the next, because if the structure went beyond that width, then you knew that it wasn't biologically accurate. So I feel like there haven't been a lot of examples of that sort of thing. And the more that there were, the more that different technology developers could use that in their benchmarking.
Yeah, I agree. I think that's really key. Do we have any other examples of, I mean, it might be nice as we're putting this together to have something beyond just brain stuff. Does anybody have any good examples or ideas of something from another tissue that might help illustrate it for folks who are working in the lung or the heart or somewhere else?
I know who published a paper on the coordinate system for the lung, where depending on whether on the main branches of airway or the sub branches, that may be needed for other organ architecture. Some are like a tree, others are like a tube. I just want to hear more about how those coordinate systems are laid out.
So we haven't published anything in this space. We've been thinking about how to build coordinate systems in the lung. I think one of the things that would make that particularly useful is if we also had molecular data that we can map into that coordinate system, but we haven't reached that point yet. Evan, to your question, the liver seems to be one of the other tissues where
Yeah.
Shalik and others have studied in quite a lot of detail, but I myself don't know exactly what would be the best biological examples to cite there. But that seems to be one where
zonation.
there are good sort of clear patterns of zonation.
Yeah. Yeah. Okay.
I have just one quick comment on, actually, I know that the KPMP or Kidney Precision Medicine team, I think they're still grappling with it, but I know that this is also very much something that they're thinking about. I'm on their advisory panel as well. And so they've been talking specifically about what they're looking more from a clinical versus diseased axis. So they're looking specifically at markers that may be associated with clinical findings of either acute or chronic kidney disease. But then in doing so, also needing to define some of the attributes that go along with cell shape and cell function that is inferred from spatial location, as well as certain properties of clinical things that have to do with kidney that I don't fully understand. But I know that they're on it, and they have whole working groups associated with trying to define some of these.
I probably want to turn to a more basic question. We just talked about fairly advanced topics for the future and how to get the spatial framework for molecular types to hang in. But more basically, it's still about technology. Most of us are coming from a new consumer's viewpoint, want to know, for my tissue slice, what is the entry-level technology and who can help us make that first decision.
All right, so you're thinking, like, from your perspective of a valuable, well, so maybe you're even thinking like some kind of like
knowledge central.
Knowledge center, yeah.
Or people who feed into it. There's a study group to update their knowledge. So those who ponder whether they should go with, say, Visin or wait for read core, or outsource it to, say, NanoStream, or just get a best person to try a home version of Merfish. Those are type of decisions are in the air and kind of affect a lot of people.
I see. I'm trying to just coalesce that into some like sort of specific recommendation, what that would look like. Do you have any other?
So in time, if we sit here and wait, in a few months, we'll be read about a review or a comparative study. But even before that timeline, is there a group to share such information?
Right. So maybe even like this sort of meta consortium could have even just it's almost like would be like a listserv or something. Mm-hmm. Are you thinking? Like where people are sort of exchanging initial impressions about the performance of things on tissues that you care about or others care about?
I guess, yeah.
Yeah. Well, I'm just trying to, yeah, I'm just thinking about how we could specifically just describe it. Mm-hmm.
So just checking in on each of the groups, we do have plenty of time left, but I would urge you to sort of try and figure out which problem or set of problems you want to develop a solution for. I've just, none of the groups have moved on to developing solutions yet. All those blue boxes are still quite a bit empty. So just a gentle nudge to feel free to put a proposal forth. Any other way I can be helpful?
No, I think what we're doing right now is we're just trying to kind of brainstorm specific, collaborative ideas, and then we would just put those in at the end.
Got it. Okay. Sounds great.
Okay. Just sort of scanning back at our notes from before. Do we want to make some kind of recommendation around multimodal measurements, trying to bring Chung-Yuan into the conversation a little bit, if he's still there? Oh, I don't see him on the list anymore. Maybe he had to leave. Yeah, what about other sort of ideas about how we could accelerate multimodal measurements? And now I mean actually like measuring epigenetic data together with transcriptional data, or are there particular challenges we think that collaboration could be useful for?
So as a reader of literature, I'm most hungry for something that helps with the comparison. For example, Evan, you publish SlideSeq, you have your own demonstration tissue. If other people publish spatial technology, if they do it on the kind of as closely as what you have done, it'll be easier for us to evaluate.
Yeah, so like just like you said, the reference tissue. I mean, that was really key in single cell, right? The fact that that this human mouse comparison, everybody started using the same 2 cell lines. And so from that same 2 cell lines, you could get both the species specificity, which was a measurement of the single cell-ness of individual profiles. And then also you got capture efficiency. So you knew the number of transcripts at each cell that was detected in each cell. So I agree. I think that is just a really, really key need in this field, is something similar like that. And it's a little bit like, I noticed that, like you said, I noticed that a lot of groups, ourselves included, are profiling the hippocampus just because it's so pretty and easy to see. So maybe that, and that sort of gets at sort of the things that we've been talking about in multiple different domains, which is like in the first part, the canonical tissue for comparison. But maybe that itself should be sort of a highlight of a very specific recommendation.
Just like the Atlas is trying to, in its first phase, building cell catalogs that have some stability as a reference. The reference map with whichever technology should aim for some semi-permanence, so the next layer of technology can refer to it.
And again, that was sort of the motivation of the Space TX project, for sure, was to do this all on the cortex, so that part of things would be kind of standardized.
Yeah.
And actually, maybe that's also a big complaint of others in the HCA community, is that all of these strategies are exhaustively deployed and validated on brain for a variety of reasons. And maybe we need to recommend that there are 4 or 5 that everybody does, right?
You mean 4 or 5 tissues?
Yes.
Yeah. So we've heard retina, hippocampus. There might be a few. Let's say.
Yeah.
Liver.
I'm just making a note of this in the blue box so that we're prepared.
Yeah. I wonder how you guys keep up with literature. I wish there is a bioRxiv channel or Slack channel that's dedicated to spatial single cell or subcellular spatial.
Well, bioRxiv does have this nice this segmenting by discipline. So certainly this field is covered pretty much exclusive. I mean, I think anything would be tagged as genomics in this one, but of course that excludes in situ based approaches that may emerge from a more microscopy based domain.
I think they have a HCA channel. I just wonder if there is a more specialized channel for spatial.
I see. I mean, what might be some particular tissues? So I think we could say, obviously, like one brain tissue. I mean, the other thing, in addition to being there, the potential need for different, organs, there's also a need for different fixation strategies, right?
Hey, so I have a question about, and thinking about different organs, whether or not it might be kind of interesting if you anchored around, or one could try to anchor around selecting a few diverse organs samples, including brain or different brain areas, all of which have some sort of vascular perfusion, because it could be kind of cool both from a scientific perspective as well as a sort of anchor point to use some perivascular structure, molecular identities. You can imagine that there's vascularization through essentially every organ. And so while you could do a little bit of discovery that's on the organ and the tissue itself, you could also essentially parse with two sort of hypotheses of what is the similarities between, say, vascular endothelial makeup or perivascular tissue composition that spans multiple different tissue types. You see what I'm saying? So like you can have almost as close as you could get to a cross organ control. Understanding that I think there is some, there's been demonstrated differentiation in terms of the actual molecular architecture of vasculature through different organs. But you've got something there at least that you could use as a little bit of a Rosetta Stone or a potential control that spans organ systems.
I'm trying to say it back. I think you're referring to some consistent landmarks for wayfinding. So when we build a canonical kind of average human heart or average human brain.
No, I mean, I think that's great and that's important as well. I'm actually talking about, again, from a spatial transcriptomics perspective. If you're looking at molecular identity, so if you've essentially got a cross section that would include some sort of vascularization, which it would be hard to miss, you can make sure that there are molecular profiles through whoever is part of the community that does this kind of mapping would be able to use across multiple different tissue types. So say, for example, it's kind of like using the only thing that might actually allow you to have some sort of biologically relevant mapping anchor point around there, and not in terms of the anatomy, but in terms of the molecular identity. So this would be just straight up spatial transcriptomics.
I think that's a really good point, Amy. I think that's a really cool idea. I think some maybe caveat is that probably you'll find that in different tissues that there are going to be subtle differences in the vasculature. I can think like liver and kidney almost certainly have differences, and probably brain too, right? So
they do, but they have some
similarities. They have enough similarity to cross-reference to. But I think it's a really great idea.
Yeah. Someone in the chat is also suggesting that the spleen might be a nice example as something that's relatively homogeneous. Also, the cells are very small, so it's quite a spatial challenge actually to resolve those tiny cells. But I also bring it up just because it's highly vascularized tissue. Yeah, that's nice. I'm trying to think about, are there other kind of like consistent features that are shared across all tissue types besides vascularization? I mean, obviously there is some degree of immune cell presence. Yeah, I just saw somebody echoed what I was saying. Immune cell content is shared, so that might be another kind of anchor point: vascularization and immune cells.
I see. So you're talking about common cell types that showed up in multiple tissue systems.
Yeah, like if you're again, yes, they're going to be examples of where these cells differ because the resident macrophages of the brain are very different from the resident macrophages of the kidney in certain ways, but there should be some consistent features, right? Shared gene expression patterns, shared and obviously the degree of sharedness is going to be different across these these different reference points. But I like that as a sort of general framework to be thinking about how to make that comparison.
Yeah. So again, just as a reader of the pan-cancer papers, they talk about how mesenchymal cells and fibroblasts or pericytes have similarity. They showed up in multiple tissues. And then even in today's talk about the respiratory pandemics, I'm interested in knowing these adult stem cells that are in every population that regenerate epithelial or mesenchymal cells. They're actually relevant to fibrotic damage or tissue repair after an infection or after some kind of damage. Turns out there are such cells. You can loosely call them adult stem cells or progenitor cells. They are, just looking at Atlas data, I've seen some in the lung, in the heart, in the liver, and in the testes. They are mappable. Yeah.
So those are cross benchmarks, but then we would obviously also, like we've said, want to identify within tissue kind of benchmarks that are very biologically driven.
I mean, that's a harder one because I think there's so much subspecialization you immediately get into, but I don't even know, like for some of the spatial transcriptomics components, if you can certainly look at like nuclear proteins. There are certain potential conserved nuclear proteins, not just the most obvious ones, but ones for which I know in the past people have looked at, which again turns into one of those slippery slopes where when you don't look at it, everybody assumes they're all the same. And then when you actually start looking at the data, it turns out to be much more heterogeneous than we thought. But yeah, there's certainly a cohort of those, as well as some cell structure proteins that I would imagine would be potentially areas to look at. Some of the metabolic, I don't actually know about some of the metabolic proteins, but or transcripts, but I imagine that would be another thing that might change from organ system to organ system.
Yeah. And yeah, so within tissue, I think we've talked about cell type, single cell defined types and evaluating their ability to be projected. And I think the other thing is evaluations about not just the sort of ability to match the types, but also evaluation of their distribution, which may be relatively well known for well-studied tissue, and their sort of proportional composition, right? And that can also be a way of, so I'm thinking like, for example, in the lung, we obviously just learned that there are these very interesting rare cell types that are embedded within more common types. And those kinds of, having a list of those kinds of examples could be very, very useful for technology evaluation and development. I mean, sometimes in tissues, like there are just sort of things that people know functionally about the tissue. So I remember like in our first retina work, when we described Dropseq, there were just certain things that people know about different behaviors of individual cells. So for example, in the retina, there are amacrine on and off cells. So these cells that respond to different patterns of light. And so, one would sort of want to know, could your technology find those particular things? You might not necessarily expect that those things are transcriptionally defined, but they might be. And there might be, for example, in that case, there are known spatial distributions of those different types. So just things where you might have kind of a priori knowledge that isn't molecular, but knowledge about, oh yeah, like, that cell type's always next to that one, or, those two things respond differently and they're kind of differently arranged. That's sort of where tissue experts are absolutely key.
Yeah.
I mean, maybe what we're thinking about is that this is some kind of like working group that sort of integrates tissue experts with technologists to sort of define a framework for doing these kind of evaluations and iterations for technology improvement as well.
Yeah, I agree. I think biologists or the physiologist can tell us more about what they know. Even in my limited interaction, I know, you see, for oocyte, the follicle is a sphere. So the R, the radius, that dimension is useful. And for tubular structure, you have the Z-axis, but you have the radial axis. And then for the liver, you have these hexagonal units, and then they're organized into, again, zonation is a word there. It has some resemblance to the apical, cortical versus the luminal dimension. So I'm sure histologists have tried to have their own terminology. We are at the opportunity to lay some to molecular features on that coordinate.
Right, right.
So how does that translate into the blue box?
That's what I'm sitting here trying. I've started putting that blue box together. I mean, I think like, look, I mean, I'm a very rule-based person, so I always like to try to follow exactly what we're supposed to do. But I think like, let's be realistic. If we could come up with even 1 to 2 kind of clear recommendations, and I really like this sort of framework of thinking about, I agree that a major weakness in trying to kind of grapple with new technology in general, but especially in this domain, which kind of has brought together people from very, very different fields, both biological and technological, is the fact that there isn't this kind of clear framework for evaluating the quality of and sort of the suitability of a particular technology. So I think if that's something that we can really expand on and give very specific, some of these nice points about, vast sort of shared features across tissue types, the kinds of ground truths that we would want to find, I think that seems like a pretty reasonable blue box entry. I'm also open, if you guys have other thoughts of things that might be useful to include besides this, we should definitely put those in as well. Anybody else have any other sort of big recommendations that?
Yeah.
Well, you had that, somebody had brought up the fixation point, sort of more of a technical, either best practices or learnings, things that are, I mean, again, I'm thinking about the Kidney Precision Medicine Project, partly because we just had an advisory council meeting, and there was a lot about that was sort of a microcosm of some of the discussion that's going through this broader group. So the intent of that whole project is to take this sort of multimodal way of bringing together a single cell kidney atlas that has all these other modes. And one of the things they came up with was working groups that were tying together the histology and the molecular profiling, in part because they found that they were able to make a huge quality improvement where they sped up their tissue fixation by a lot. So some of us who work with RNA all the time are like, well, of course, but the histology people hadn't really known that because they didn't need to worry about that for what they were doing before.
Yeah.
And so that learning, when it happened, just made this really huge difference in terms of the quality of data. And it was this big like aha moment for everybody. And that was through the sort of iterative working group that they had that was established within that consortium. So if you think of that generalizing over HCA and HubMap of, okay, getting together people that have specialization, thinking about the tissue specific qualities practically, both in terms of morphology, just tissue type best practices that come through histology or tissue handling, and then pairing that up with some sort of like molecular anatomy kind of perspective of what do we know about the RNA and the features that we tend to see in these cell types. That could be really powerful. I think that would be an amazing output. Very blue boxable.
Yeah. I like to echo what Amy said. I really think this is related to, there is a later breakout session on cell ontology. We are talking about a proper spatial ontology for cell types and for different organ structure. We want to kind of harden what's been done before, but have a proper zip coding system. You know, in zip code, some digits are for coarser location, others for finer location. Different geometry requires different longitude and altitude to get that working group thinking this way, so that we will come to the day when everybody's publication, you get a picture, either it's 2D or 3D, whether it's a small patch or big patch, you need to reference to that some global waypoint so that data can integrate in that spatial way. Otherwise, everybody just publish their own stack of slides. We don't know where they are.
So I added both to the document as a comment as well as to the chat here, just a link to a recent bioRxiv article that the Kidney Precision Medicine team has, and it's not 100% relevant to this, but it's relevant in the sense that it pulls together some of the different data standards and quality standards that they've found to be really important early stage in building up reference points and sort of quality accountability cross project. So that's forcing them to work with clinical data as well as the molecular profiling data, as well as just some of the certainly transcriptomics data and kind of the storming, forming, norming process that went through that. And it might just be useful for, if you have time, anybody who's interested in kind of what that looked like, because it also speaks to what Juna just said about needing to sort of reference a framework for the context in which you're speaking of something. Right. So this isn't about, the way it's presented isn't so much about a paper that gives the findings of all the data, but it gives sort of the findings of how people are going about to get the data. And in that context, it may just be kind of an interesting reference point.
Yeah. So that will eventually become requisite metadata when somebody is publishing a tissue characterization paper. I remember the day when we have a neuroanatomist pointing out a brain saying, that's hypothalamus, cut that. We don't know, we just cut it, but we videotaped it. So there is some accountability. In the future, it would be nice to have this kind of shared accountability on where things are.
Yeah. I mean, that can also kind of fit into this canonical tissue thing, right? Where we
yeah, that's right.
Yeah. Just making a note of that.
Yeah. It also goes back to that older, every decade you see some kind of a virtual human project, or the average soldier project. Like
that. I'm also thinking about this fixation thing. I mean, one of the challenges with human tissues is that they're very much under the control of pathologists, and pathologists have often been doing the same. Have been sort of preserving tissue in the same way for a long time, and it works very, very well for their purposes. I wonder if it might be valuable, and I don't know if such a thing exists, but I wonder if it might be valuable to have some sort of like meeting or forum in which those who are handling these tissues and procuring these tissues can interact more more intensively with those who are making, the biologists who are making measurements on the tissues for research purposes, and also technologists who are trying to build tools to see what kinds of, ways there might be to kind of diversify the kinds of fixation strategies we can take, or, kind of get us to that point that Amy mentioned for kidney of, streamlining an approach that's yields better data for certain modalities. I think that could be very valuable.
By fixation, you mean still the landmark finding to annotate location?
I mean that basically almost all tissue that comes out of a person's body is being, formalin fixed and then paraffin embedded, right?
Yeah.
And that basically closes the door on almost every genomics assay there is, right? And you hear all kinds of things. You go to places, people say, well, I had somebody's telling me that they've figured out that if you just heat the tissue in advance of formalin fixing it, that dramatically improves RNA quality, for example. I have a feeling that there's a lot of things that could be tried in terms of the process by which tissue comes from the patient in the OR and then reaches the researcher, both in terms of chemicals, in terms of the time frame, that potentially if there was sort of if there was a larger scale effort that was in which many people were participating, could actually be very, very powerful. Because I think that that's a major limitation right now.
It's like a blue box thing.
Yeah, it sounds blue boxy. I don't know.
Yeah, sorry, it's Christina. I would add that people have even published some protocols that try and adapt this. Unfortunately, the challenging part is getting the uptake amongst pathologists, around the world as an SOP. But I do think it's an important discussion. I think equally when we're not fixing, I know as part of HCA and the HCAN efforts, there is a lot of fresh tissue collection, but that suffers its own challenges, and it does differ by tissue type, and it depends on whether it's healthy or malignant. There's a lot of factors that play into that. But I do think that best practices there and discussions are happening to some extent, and maybe that's part of a broader dialogue here that could happen.
Yeah. And I think this question about like tissue time, for example, ex vivo time is totally key. We have in our own work have observed in the brain. We obtained some samples from surgical samples or biopsy samples, and there's just for profiling purposes, there's just a profound quality drop even within minutes of protocols that take minutes versus tens of minutes to reach freezing, for example. So I think there's a lot of different aspects of a sort of procurement pipeline, and this has been discussed a little bit in the HCA, but I think it's been really the focus has really been on the single cell aspect of things, because that's been what we've been doing the most measurements on. But I think we have to increasingly be thinking about it from the perspective of these different in situ modalities.
Yeah. Another consortium where we do multi-omics, there is intense discussion about who gets the first cut. We know metabolites can change depending on whether you do bloodletting first or not. And then phosphoprotein people are the most worried about oxidation. They want to get the first sample within minutes.
Yeah. Any other blue box thoughts?
So Evan, if you look at the chat, there are some comments.
Oh, okay. Somebody said, I wonder what will be the way to share the spatial data sets.
Right.
Yeah. This is something that the community has been starting to really work on. CCI and HCA are thinking about sort of strategies for depositing the data so that we have flexibility to view both the digital data, processed data, but also the unprocessed data. I think that is definitely key.
I gave a link just to what we have running right now at the Allen Institute for some histological data. So you'll notice this isn't spatial transcriptomics data yet. It's not SpaceTX data because actually CZI is the one that's working on the I think, the viewer and the data analysis visualization for that.
Yeah.
But I bring this up based on an earlier point of just how to, questions about how to share data. We had this browser that we created years ago now. It's not the best or the fanciest, but the intent was to try to create just a way to visualize accompanying histology for the single cells that we were profiling. So, this is again in brain, and this is just what we did in a sort of isolated way. This specific thing isn't necessarily scalable or extractable to every scenario, but it's just an example that's there. And it's also a little kludgy because the way you access the data, you have to like click on a specific column. That's the donor column. I don't think it's intuitive, but given the budget we had to work with at the time for getting this out the door, this is what we've got right now. But you can, if you work with it, you can find histological images that you can download or look at in a browser. And this is just an example of making the data available. So just as a reference point for people.
Yeah.
So Amy, these are histology on the adjacent or flanking slice, right?
So these are a little of both, so to speak. So you'd have to look at, if you go to the technical white papers, which are in that main page, it gives a whole description of the methodology for acquiring it. So there's a sort of interleaving of histology that we do for when we record from single cells and fill them. So yes, it is adjacent slices, but there's no reason why it would have to be, if that makes sense. It could be done in a way where it's not. For this particular experimental paradigm that we were using for this, made sense for adjacent slices because we were using actually a different piece of the tissue to acquire the single cells that we were doing in bulk. Yeah. So, but again, I'm using this as the example for making the data available. How do you use it? How do you see it? There it is. This is an example of making it available. That's actually separate from the publication. So there's a separate publication that sort of describes broadly what this data is, and there's actually a few publications. But this is just kind of a way to allow people to see it and navigate it.
It's image as part of the sample metadata. Very good.
Okay. I think we've done some pretty good work. I think so. I mean, we can fill out this. I think I can easily synthesize. If anybody wants to just have a look at this blue box and put anything else they want in it, but I think, to me, I think it's a nice output is particularly this question about sort of thinking clearly about the tissues that will be very defining for measuring and inventing new technologies. I think that's a very powerful recommendation. And it's actually not very specific to this subgroup in the sense that it's not any more related to anything than, to sequencing based metrics than to in situ based metrics. But I think it's something that the field really needs. Happy for us to end a few minutes early, or if you have any people have any other comments or thoughts.
Evan, I want to ask a new question. There is a concept of housekeeping gene, something invariant in every cell. But here, we'd love to have, by the same spirit, a panel of genes that are fairly universal. So some always mark a nuclei, some always mark a membrane. So there is some consistency across the largest number of cell types that's easy to measure that can be pushed onto most of the emerging platforms.
I think that might be more relevant to protein, which has such, I think for RNA, there are obviously RNAs that live in the nucleus, and there are some like in the brain that get trafficked out, but they're pretty few and far between. I mean, most RNAs are just kind of sitting in the perinuclear ribosomes being translated. So I don't know if the subcellular localization of RNA, I mean, I think it's a great in particular metric, but I think it's a little bit of a like niche metric. I don't think it's the thing that most people care about when they're looking at transcripts, but for protein, it's obviously really key.
Yeah. So I'm mainly thinking about a subset that's the most boring. It's a structural protein don't go up and down with cellular state or regulation, but it's great for, know where you are, in which structure you are.
Yeah. Mm-hmm.
Andrew, I have a random question to throw out. Maybe it relates more to protein, but anyone thinking about extracellular material? Stuff, cytokines that are being secreted, if you're interested in like paracrine interactions or ECM, and I mean, we're missing all of that, right?
No, I totally agree with that. I agree. It's more of a protein question. There are exosomes and things like that probably are kind of orbiting around. I think a lot of that stuff gets washed away when you, when you prepare the tissue, you slice and freeze it. I mean, it depends on what you're doing, but if you're doing any kind of fixation process, I think a lot of that stuff doesn't get fixed very well because it's sort of floating around in the extracellular matrix.